reduced and the substrate. Okay. So it's a chronic disease characterized by elevated level of glucose, blood glucose, leads serious damage to the heart, blood vessels, eyes, kidneys, and nerves. And according to WHO, the diabetic is seventh death-causing disease. Recently, it is declared that it is the seventh death-causing disease in the world. In 1921, so Nobel Prize was awarded to F.T. Banting and J.J. Richard McLeod in 1923 in Physiology and Medicine for discovering insulin. And since then, the research work started on diabetics. In 1930, discovered diabetic and insulin dependent and non-insulin dependent, these two. And before that, we had only the diabetic or the sugar disease or sugar related disease we used to call. And from 1930 onwards, we started calling this in two divisions. One is the insulin dependent, another one is the non-insulin dependent one. And there are several factors. Initially, people thought that must be there are the genetic factors responsible, but later on in the last almost 80, 90 years, the researchers understood that more than genetic factors, the non-genetic factors are more important one. And we'll see what are all those important factors. So this, as I told, the disease is divided into two groups. One is the insulin production, other one is the glucose entry into the cell. Two steps are there. And if both the steps are clear and normal, then there will not be any disease. If any of the step is disturbed, then there will be the disease. So, and we can divide this based on these two in two types. The one type, diabetic 1 is in diabetic 1, so insulin is completely not produced, or sometimes very little insulin produced, and it is completely insulin dependent. So we have to have insulin injections every day, then only the patient survives. Otherwise, there is no chance of in survival. Similarly, the other one, type 2, where the insulin is produced, but the insulin is not associated, entered into the cell. There are several reasons here, not one or two. Even now also, after 90 years, we are not in a position to understand or tell exactly that these are the reasons for this particular mechanism. Then we have the diabetic insipidus. This is another abnormalities we have, and this
Hello, sir. Vidyasagar, sir. Hello.
We can't hear. Maybe some network issues.
Yes, network issues. Can wait for 2 minutes. Sir, Vishwasagar, sir. Vidyasagar, sir. Sir, Vidya, sir. ಸರ್ ಸರ್ ಅನ್ಮ್ಯೂಟ್ ಆಗಿದೆ ನಿಮ್ದು. ವಿದ್ಯಾಸಾಗರ್ ಸರ್, ಸರ್ ಕೇಳಿಸ್ತಾ ಇದೆಯಾ ನಾ ಮಾತಾಡೋದು?
ಕೇಳಿಸ್ತಾ ಇದೆ, ನಿಮ್ಗೆ ಕೇಳಿಸ್ತಾ ಇದೆ, ನನ್ ಕೇಳಿಸ್ತಾ ಇಲ್ಲ ನಿಮ್ಗೆ ಅನ್ಸುತ್ತೆ.
ಇವಾಗ ಕೇಳಿಸ್ತಾ ಇದೆ ಸರ್.
ಓಕೆ, ಈಗ ಕೇಳಿಸ್ತಾ ಇದೆಯಾ? ಅಂಡ್ ಇದು PPT ನೋಡು, ಇದು ಇದೆಯಾ?
ಹಾ ಸರ್, ಫುಲ್ ಸ್ಕ್ರೀನ್ ಮಾಡಿ.
ಓಕೆ, ಮಾಡ್ತೇನೆ.
ಸ್ಲೈಡ್ ಶೋ.
Is it okay?
ಓಕೆ ಸರ್, ಓಕೆ, ಕಂಟಿನ್ಯೂ ಸರ್.
ಇಲ್ಲಿ ಪವರ್ ಕಟ್ ಆಗಿತ್ತು ಇಲ್ಲಿ. ಓಕೆ, so the diabetic, as I told, there are two stages, very important stages. One is insulin production, other one is glucose entry into the cell. And the insulin production is the most important one seen in type 1 diabetic, and the second one is very common in the diabetic 2, where the imbalance in glucose entry is due to the, I think, resistant and many other mechanisms. Insulin is resistant or other several mechanisms involved in the cell. And because of that reason, glucose will not enter into the cell, and that results into type 2 diabetic. And another one is the diabetic insipidus. Insipidus is another type of diabetic, which is very rarely seen, where the kidney cannot concentrate urine properly. Body is not able to retain water and produce more diluted urine in this one. And the most important hormone is synthesized here, that's vasopressin, which is not in this one. Hormone is not properly synthesized, and disease is seen. So this is the very clear picture. In normal case, we can see here the white dot stars are the glucose, and the yellow stars are the insulin. In normal case, insulin binds with the glucose and take the glucose into the cell. Whereas in type 1 case, there is some dark stars, and these dark stars are not the insulin, but they are the fatty molecules. And here the insulin is completely absent, therefore the glucose is not able to enter into the cell. Therefore, the glucose concentration increase in the blood vessels or in blood, and diabetic will be very serious in this case. Type 2, here we have in the blood vessels, we have sugar molecules. Along with sugar molecules, we have also the insulin, and see the rate of insulin here. Insulin is more as compared to the normal, and glucose is carrying or entering into the cells, but the level is very, compared to normal one, the glucose entry into the cell is limited only. Because of more amount of insulin here, so there is a possibility, or the cell resist the entry of insulin and glucose in this one, and that results into resistance. Insulin resistance develops the later stage, and there will be deficiency of glucose in the cell. So these three pictures clearly says that how the diabetic develops and how the sugar enters into the cell. Other than this, we have gestational diabetic, maturity onset diabetics, and neonatal diabetics. These are mitochondrial diabetics, cystic fibrosis related diabetics. These are also almost not that common, but okay, these are also available. In very few cases, we see these diseases. The first one is associated with the pregnancy. Second one is associated, it is with the genetic defect, that beta cells functions. And third one is almost, it is a for a very short period, this will be seen in the children. And third one is associated with the mitochondrial dysfunctions, which is almost resulted due to non-autoimmune failures in the mitochondria. And last one is due to insulin deficiency, but insulin resistance also seen during the disease. Second diabetic, which is very predominant, increasing dramatically all over the world. And today, it's almost about 150 million people worldwide are affected, and majority are from the lower middle income countries. Then almost total 1.5 million deaths occurs annually, and which attribute directly to the diabetics. And of these deaths, for almost about 70 to 80% are due to heart attack. And India is almost today in the topmost position. Earlier China was there, but now India is in the topmost position. According to ICMR studies published in 2023, we have today almost 10 crores population with diabetics in our country. And there is also a report which says that the incidence of diabetic in some metro cities, say for example in Kolkata it is 11.7, and in Kashmir Valley it is 6.1, and in New Delhi 11.6, Mumbai 9.3, and Chennai 13.5, Hyderabad 16.6, and in Bangalore it is around 12.4. So this picture or these studies indicate that is leading the diabetic as compared to other north and the hilly regions of India. So don't know the reasons behind all these, but the picture shows that the South India is more predominant for diabetic, and that too the Hyderabad. Then incomplete oxidation of fatty acid contributes to insulin resistance in heart and muscles. So this is another important factors. This is the picture which gives us clear the understanding for how the diabetic blood circulation occurs. See, when the blood sugar is more in the blood, then it enters into pancreas. Then pancreas produce insulin. Then insulin associates with the sugar and take it into the cell tissues. And when it is metabolized and used, then the sugar content in the cell comes down. And during that time, the low sugar condition again, in the low sugar contents, the glucagon releases from the pancreas, and then the glucose converts, and that glucose again used, and the condition maintains. So that means we have two enzymes here. One is the insulin, another one is the glucagon. The hormones, these two hormones, the insulin associates with the glucose, take the glucose into the tissues, cells, and utilize it. In emergency, whenever the glucose condition comes down, then we have glycogens in the liver, and this glycogen will be converted into glucose by glucagon hormone again, and again this glucose will be used in the metabolic reactions. So this way the glucose circulates in the blood and tissues of different organs and maintains the glucose level in the blood. So pancreas we have here. So alpha cells and beta cells are very important in the islets of Langerhans, and these beta cells are producing the insulin, and alpha cells are producing the another hormone, glucagon. So diagnosis, how the disease is diagnosed, as we all know that in the normal case 70 to 100 mg per dilution is the normal fasting one, and in the after meals the limit is almost 140 mg. So above 140 mg will be considered as pre-diabetic or sometimes diabetic. People now, even after 130 or 140 mg sugar, so they consider, some doctors say that it is diabetic and just medications. And in real sense, so we have some studies, reports I have seen from different papers, recent papers, that more brain cells die in the low blood sugar. The mortality rate is 15% at HbA1c between 7 to 8. 7 to 8 HbA1c is considered as the diabetic one as per the current situation. But during this situation or in this condition, the mortality rate is less. Whereas less than 7 and more than 9 HbA1c, the mortality rate is high. That means the HbA1c should be between 7 to 8. Even if the doctor says that it is diabetic, that is not diabetic, it is a normal situation where mortality or cell death rate will be less. And these are the symptoms we find always in the diabetic patients, that polyuria, more urination, the polydipsia, more thirst, and polyphagia, that's more hunger, and blurring of vision, weight loss, and ketonuria. So these are some of the symptoms we find in the diabetic patients. Now it is understood that the blood sugar up to 140 is not the diabetic one, and above 140 to up to 201 is the pre-diabetic one, and above 200 is the diabetic one. But even this also, as per our system, Ayurvedic system, the blood component or blood concentration and the sugar concentration in the blood varies. It is dynamic, varies from time to time. Since morning to evening, if we take the data, there will be lots of variations or differences. In the morning, we will have different, in the afternoon, evening, and all this. So we cannot consider this data as a stable data and measure the situation or stage of the diabetes. And it varies from person to person. Every person is not similar in sensitivity of the glucose. There will be some variations. So therefore, particularly in Indian populations, as we have different ethnic populations, we have different food culture, we have different geographic conditions, we have different genetic makeup as well. So therefore, the same rule which is followed in other countries or in foreign countries, Western countries, not be applied to Indian populations. So therefore, as on now, if any symptoms, these symptoms like polyuria or polydipsia or polyphagia, if any of these symptoms are seen, then probably we can go to the doctor and see what is the situation, and then we can have some kind of medication or precautions. So here, the causes, some of the most important cause is food, socioeconomic status, regional imbalances, pollutants, ecosystem, sedentary lifestyle, and genetics. So these are the factors which induce physiology and psychological abnormalities, and that results into stress, hypertension, obesity, digestive disorders. Food, almost in Indian culture, as I said earlier also, we have different ethnic population, we have different geographical conditions, temperature, humidity, water, food habits, everything is different. And we cannot use one particular food throughout country in different locations. So we have different food in different specific food for specific location. Similarly, the conditions also, geographical conditions also different. So therefore, so we are missing here more diabetic is only, most important is one. We are not choosing the specific food which is required to for a particular population living in a particular geographic conditions. This is most important. Second important one is gut ecosystem. The gut ecosystem these days is almost lots of studies are going on in gut ecosystem, gut microbes or gut flora, we say, where almost 90% populations are not updated for gut ecosystem today. The reason behind, again, the food, specific foods we are not specific, that means the regionally important foods we are not consuming. Western culture, Western foods we are adopting, and probably in that situation, the specific microbes are not as colonizing in the gut. And as we all know, we have only around 23,000 genes in our cell, and we have millions of microbes in our gut. If the gut flora, gut genes are more than the human genes. Therefore, our body is almost belongs to microbes more than our parental genes. So we need to concentrate, or we need to understand the importance of gut microbes, and we have to feed them equally, and then only they can serve. They can serve for us, and particularly health. Then another one is the genetic disorders. There are several genes involved in this metabolisms. Lots of genes, even in the last 80 years, we have only a few genes have been identified. Although there are several genes, only few genes have been identified that they are the important one, and still many things are incomplete. So therefore, we cannot say that how many genes are involved. In one person, one type of gene group is involved. In another person, another type of group of genes are involved. It's very difficult at this moment to say that these are the only genes involved in the development of diabetics. Hypertension, stress, obesity, digestive disorders, all these are very, very important ones related to this disease. Second important component is the drugs. Antibiotics, particularly pentamidine, nicotinic acid, and so many other steroids we are using these days to control the microbes or for many disease disorders. And one or other way, these are interacting with the beta cells and disturbing the digestion or disturbing the production of the insulin in the body, and that is resulting in the formation of diabetics. Then abnormalities in the pancreas, again, due to so many infections or so many abnormalities in the psychological or physiological conditions. In spite of all this, so we are still we are not in a position to say that these are the exact cause of the pancreatic failure or the insulin resistance. It's unclear. So genetic disorders, about 10 loci in the human genome have been identified in diabetic 1 case. 10 genes have been identified, although there are many other genes, but these genes are confirmed that these are involved in the type 1 gene at the locus on chromosome number 11 and chromosome number 7. So these two genes have been identified and studied, and still many other genes are underwork. There is one more gene, that's KLF11, which regulates insulin gene expressions through binding to the insulin promoter. The first picture is the KLF gene structure. See, there are four exons, and there are different sites in this. And this is the transcription factor. This gene regulates a protein, and this protein acts as a transcription factor, which binds on the insulin gene promoter. In the second picture, there is a promoter, that's the insulin gene promoter here, CCCC GCCCT. This is the location where this transcription factor binds and carry out the gene regulations of insulin. Suppose if this KLF11 is normal, then it binds on this particular promoter, and there will be the gene regulation and insulin production will be normal. But now some mutants have been identified in different exons in the KLF11 gene, and these mutants are not able to bind down the insulin promoter. And if it is not binding on insulin production, then insulin will not be produced in the cell. So this way, this is very, very important gene which has been identified and the mechanism. And there are the several mutants also identified, and some mutants are capable of binding with this and producing the insulin, but the rate of insulin production is lesser than the wild. That means if the mutation rate is more, then the insulin production will be less. So this way, the genes are acting. here the PGC1 alpha, another gene is identified, which is emerged as the key regulator of several aspects of mammalian energy metabolism. PGC1 alpha is a very, very important one. And again, this is also a cofactor. It's a cofactor one. It's a small polypeptide which acts as the cofactor during the regulation. It binds with the transcription factor and enhance the production or enhance the regulation of several mammalian genes, particularly the genes related to metabolism. Recent studies shown that the reduced expression of PGC1 alpha in skeletal muscle of diabetic and pre-diabetic humans. So this, if the expression of this gene is reduced, then probably this is one of the important hint or the sign that the person or the patient is going to have the diabetic conditions. So based on this expression, we can identify the status of the persons and we can screen out the population whether this gene is passing or is there in the genome or not. And based on that, we can identify the pre-diabetic cases or the diabetic cases and take precautions and to avoid the severity of the diabetics in the days to come. And it is intimately involved in disorders such as obesity, diabetes, and cardiomyopathy. So therefore, this is very, very important to screen one. And now the facilities are available. People are going for the screening, and probably this will become shortly very common in the days to come. So complications involved here are the cardiovascular and cerebrovascular, that is neuropathy, then Alzheimer's, the renal, kidney disorders, eyesight, and immunity. These five important disorders are associated with the diabetes. Then coming to pathophysiology. All this part is the introductory part. Now the pathophysiology components involves, it's a branch of studies related to pathology and physiology, or the ordered physiology, or the physiology of a disease cells, we can say, or disorder cells. And the main component here in the diabetic, as we have seen, is the hyperglycemia, which generate reactive oxygen. This is important. Since many years, people understood that the ROS, reactive oxygen species, are very, very important. They generate in presence of more glucose in the cell and cause lipid peroxidation and membrane damage, which leads insulin resistance, disruptions of beta cells function, and peripheral neuropathy, cardiomyopathy, mitochondrial dysfunctions, nephropathy, and foot ulcers. So most important one is the reactive species, oxygen species, people understood. But now there are other things are also experienced or identified along with the ROS. But ROS stands important even today also, which is very, very, and now it's very interesting to understand the presence of reactive oxygen species. So we have these two types of disorders, micro and macro. So micro disorders are almost the like nerve damage, kidney disease, eye related issues, and macro disorders are the heart, stroke, vascular issues, and foot syndromes and all. So this we can study in three components or three parts. Insulin is the key factor in diabetic, and we can study this in three groups. One is the effect of insulin on glucose metabolism. Another one is the effect of insulin on lipid metabolism, and third one, we can study the effect of insulin on proteins. So these are the disorders, some of the complications, as we saw in the last slide, that the wound in foot, that won't heal, leading to amputation, then the heart disease, then the blood vessels, if they are damaged, in retina, which can cause the blindness and the stroke, kidney failure. So all these are the disorders which are seen in the patients. So this okay. Effect of insulin on metabolism, glucose metabolism. See, insulin plays a crucial role, as we have seen, in glucose metabolism by facilitating the uptake, utilization, and storage of glucose in various tissues throughout the body. The first one is the glucose uptake. Glucose uptake, where insulin promotes the uptake of glucose into cell, especially in muscle cells and fat and liver cells. It does this by increasing the translocation of glucose proteins, particularly some specific proteins, and to membranes, which allows glucose to enter the cell. Then glycogen synthesis, it occurs in the liver and muscle cells. Insulin stimulates the conversion of excess glucose into glycogen through the process of glycogenesis, and glycogen serves as the storage form of glucose and can quickly broken down into glucose when it is required. Then third one is inhibition of glycogenesis. The inhibition here, the breakdown of glycogen into glucose, known as the glycogenesis, primarily in the liver. This action helps maintain blood glucose level within the normal range. Sometimes the inhibition of glucogenesis is also important. Gluconeogenesis. Inhibition of gluconeogenesis. Insulin suppresses the gluconeogenesis, the synthesis of glucose from non-carbohydrate precursors such as amino acids and glycerols, primarily in the liver. By inhibiting gluconeogenesis, insulin further helps regulate glucose levels. And next one is the promotion of lipogenesis. Insulin stimulates the conversion of excess glucose into fatty acids through a process called lipogenesis. Then another last one is the inhibition of lipolysis. Insulin inhibits the breakdown of stored triglycerides into fatty acids and glycerol, known as lipolysis. Then dysfunctional wound healing. So here we have different stages of wound healing. So macrophage, macrophage. So wound generally in the diabetic cases is not healing properly. There are several reasons for this. The most important reason is the macrophage plasticity. Macrophage is one of the most important component of blood, which has got lots of plasticity. It changes with different environments, internal environment particularly, and act differently. In this case, when in presence of more sugar, so it attains one stage and it remains stable in one stage, that is pro-inflammatory stage, CD14 plus and CD16 minus. This is the pro-inflammatory stage where this particular stage of the macrophage remains for a longer period and continues inflammations. Then its other stage is anti-inflammatory. It also produce the cytokines which are anti-inflammatory in nature, and the structure of macrophage here is changed. Look, change, and that structure, so we call that CD14 plus and CD16 plus. And the reduced anti-inflammatory genes expression also very, very important. During this stage in diabetic, when glucose level is increased, the anti-inflammatory gene expression is reduced. Similarly, inflammation by neutrophils also reduced when there is more glucose in the cell. Then cellular hypoxia means more oxygen is used in the particular region, and deficiency oxygen is seen, and because of the deficiency of oxygen, probably most of the mechanisms, the healing mechanisms are disturbed. Another important is because of the glucose, more sugar here, microbial contamination, the microbial colonization also increases. Now, recent study says that along with the bacterial populations, even the fungal populations also associated with this and made this healing process very complex. Some microbes, some fungi, for example, Candida, are associated with the bacteria and made the healing process very, very complex one. Then extracellular matrices, which are essential for healing the process, but here because of the more sugar, the extracellular matrices are not synthesized properly, and the healing process is delayed. The last one is the epigenetic basis, which is very, very important in healing this process. So epigenetic means it is almost the methylation, DNA methylation and histone modification are occurring in the genes, and this methylation process is completely based on the conditions situated, conditions available during that particular period. Epigenetic means almost it is not the genetic changes, so the external factors or internal factors are influencing the gene regulations. So whatever the factors available, say for example, more amount of glucose is one of the factors, and some biochemical changes, abnormal changes occurs, and those abnormal component of the blood and component of cells are activating some gene and suppressing some other important genes which are essential for healing process. And that is because of that, the healing process is delayed. So effect of insulin on metabolism, lipid metabolisms by promoting lipid synthesis, storage, and inhibiting lipolysis. Here is the breakdown of the effect of insulin on lipid metabolisms. So here there are the 4 stages: lipogenesis, triglyceraldehyde synthesis, inhibition of lipolysis, uptake of fatty acids, influence of lipoprotein lipase. So then there are again the insulin impact on the regulation of protein metabolism by influencing protein synthesis, breakdown of the proteins, and synthesis of amino acids or utilization of amino acids. And there are 4 stages: protein synthesis, inhibition of protein breakdown, amino acid uptake, and utilization, regulation of gluconeogenesis. Then mitochondrial dysfunctions is increasingly recognized as the significant in the contributions of pathogenesis and complications of diabetes. So mitochondria is one of the most important component of cell where the glucose metabolism is carried out. Lots of enzymes or genes are involved in this process. Any of these genes or any of the enzyme is affected, then probably the process is disturbed, and that results into abnormalities in the cell. And this abnormality in the cell creates the ROS synthesis, reactive oxygen species, and reactive oxygen species if it is increased in the cell, then there will be disturbance in all the mechanisms, particularly the enzyme biochemical reactions, and abnormalities seen. So that is the one of the reasons why that we have to have the balanced diets. We have to have particularly antioxidants from the plants are used. If the antioxidants from plants are used, then the reactive oxygen species reduction will be there, and somehow the stress in the cell, cellular stress we can reduce. And if the cellular stress is reduced, then probably the all the mechanisms we can bring to the normal state. Then another component is the pharmacology. In pharmacology, we study the interactions of drug with the cell, which is very, very important in the treatment. And now, as I told, the ROS molecules are generating more in the diabetic cells because of the abnormalities in the pathways. The antioxidants plays very, very important role to protect the beta cells. There are different types of antioxidants. Antioxidants in plants, for example, tannins, flavonoids, vitamin C, vitamin E, even vitamin D recently identified that it plays very important role in the diabetic management of diabetic and function beta cell functioning and prevent diabetic induced ROS formations. Then we have polyphenols as flavonoids, tannins, which inhibits hydrolytic and oxidative enzymes and known to have free radical stem anti-inflammatory and anti-diabetic properties. We have right now available therapy for diabetics. Few drugs, synthetic drugs are available. Successful treatment has yet to be discovered. So for diabetic, I think even after 80 years of this occurrence of the disease, we are not able to identify or produce a most successful treatment or a drug to treat the diabetic. The reason behind it is a very complex one. It's not that simple. It's not because of one or two causes. Several agents are involved in causing the disease. Therefore, still we are not able to identify the proper drug. However, insulin therapy and oral hypoglycemic agents are the first line of treatment right now people are using. Currently, there are 6 classes of oral anti-diabetic drugs, and these are the drugs which are generally prescribed in the prescribed. Among these, the one which is metformin is considered as the important one, which has shown very positive and very few negative impact on cell in this one. Whereas the other drugs, although they are reducing the sugar, bringing down the diabetes, but there are lots of side effects involved in this drug. Then we have herbal formulations. We have some formulations available in the market, and these are the Diabecon, Thiasulin, pancreatic tonics 180. Then Chakrapani is also available. So bitter gourd powder and many other drugs are available. The herbal drugs are available in the market. So coming to the plants, medicinal plants or ethnobotany or ethnomedicines, almost 1,200 ethnomedicinal plants have been identified, which are useful in the treatment of or in the management of diabetics throughout the world. And out of this, around 200 anti-diabetic compounds have been isolated or they are under trial. They are alkaloids, carbohydrates, glycosides, flavonoids, steroids, terpenoids, peptides, amino acid, lipids, phenolics, etc. Some of the plants, only few plants I have given here: curcuma, momordica, allium sativum, aloe vera, phenospora, gymnema, ocimum, and Catranthus rosea. So only few plants I have given, but since the disease is metabolic disease and involves lots of metabolic reactions. So not only these, almost every plant is involved, every plant is responsible, or one or other way it is bringing down the diabetes. So therefore, if we are consuming plants, different plants or different vegetables in the form of either food or in the form of drug, probably they have more or less efficacy or activity, anti-diabetic activity, and they will be useful in managing the diabetes or sugar disease. Curcuma. So curcumin is exhibited anti-inflammatory and antioxidant, anti-apoptosis properties. It has been shown to improve insulin sensitivity, enhance glucose uptake, and protect pancreatic usage. This is a very important one. So curcumin in 2 forms, enol forms and keto forms, and both are equally important. They are doing all this this one working very good in the diabetic, along with many other properties like antioxidants or anti-inflammation. So they are also protecting the pancreatic cells or beta cells. So recently, clinical trials and preclinical studies have shown that curcumin exert protective effects against a variety of diseases, including diabetics and cardiovascular complications. So therefore, this plant is one of the most important plants today for diabetic and other disorders. Totally, the cell health is improved with curcumin. Then another plant is Gymnema sylvester. Gymnema sylvester is a very common plant, particularly in our area in Hyderabad, Karnataka, this Gulbarga region. Plant is available, and many people are using this as one of the important drug for diabetic and even some others. In my laboratory also, we have worked on this plant. We have isolated some active molecules. We have also isolated some active molecules, very effective active molecules from the fungal endophytes of this plant. We isolated almost 10 endophytes, fungal endophytes, of which 3 endophytes were found to be very, very effective. They are producing some compounds. We have not yet published those results, but those are even compared to the available drugs in the market. So these compounds are effective. Along with the diabetic conditions, they also protect the cells and damaging cells particularly, and also they reduce the damages in the liver. That means hepatoprotective properties are seen in these compounds. So here we have gymnemic acid 1, 2, 3, and also 4, which inhibit the absorption of glucose by intestinal receptors and decrease postprandial glucose level. Then we have Momordica species. As we all know, those people are using these days this Momordica regularly. The diabetic people, anti-diabetic, anti-inflammatory, antioxidants. It has got antiviral and also anti-cancer properties. First time these compounds isolated in 1966 as a mixture of 2 polystyrol glycosides, and now it is isolated. Pure compounds have been isolated, and they are now they can be used for the treatment of diabetes. It has got some other compounds like Charancia, polypeptides, and Design have demonstrated hypoglycemic effects by improving insulin sensitivity and promoting glucose uptake. So this is the way this plant, Charancia, improves glucose tolerance, induce glucose intake, enhance insulin secretion, then activate several pathways, not only AMPK, and suppress postprandial hyperglycemia after food, whatever the glucose level increases, that also it is suppressed, and stimulate adiponectin release and protect islets of beta cells. Then Allium sativum. So another very common plant, cardiovascular. It is very, very important for heart disorders, because it is people are using this particular plant as the blood thinner, Allium sativum, because of presence of allicin. And in addition to this property, it is also used in reducing the diabetic condition, which enhance the insulin secretion and reduce insulin resistance. Another plant is aloe vera. So aloe vera, we have got several features of this plant, properties of this plant for other purposes, but also It is very interesting that it has got immunomodulators, very key immunomodulators present in this plant, and these play important role in insulin resistance. Then we have another plant, Tinospora, which is very commonly used in diabetic and heart disorders. This also plant has shown some compounds like berberine, which improves insulin sensitivity, reduce glucose production in liver, and enhance glucose uptake in peripheral tissues. Osimum, there are some compounds, flavonoids, triterpenes, saponins, essential oils, and these are one or other way involved in reducing the sugar conditions. Similarly, the Catranthus rosae plant, which has got already known, so vincristine, vinblastine, anti-cancer and drugs, they are proved to be anti-cancers, but also they have got the property, glucose uptake properties in peripheral tissues, and which modulate insulin signaling. This modulation is very important in immune modulation particularly. And so now in the features, people are particularly in diabetic 1, the beta cells are getting destroyed completely. So no medicine is available, or no medicine is working over the type 1 diabetes. So therefore, now in some laboratories, in some Western countries, universities and research institutes, research is going on the regeneration of human beta cells. So human beta cells and some other animal beta cells are also have been identified, and replacement of beta cells are taking place probably in another few years. So this also may come in the practice. Then brown fat transplants. So brown fats, adipose tissues, we have adipose white fat is there, and brown fat is there in the
কোথায় গেল তো ক্লিপ যা ক্লিপ কোথায় গেল
hello. Hello. Okay. Hello. Can you hear?
Nothing, sir. You can continue.
Okay. And again the epigenetics one. So epigenetic mechanisms regulate gene expressions via various forms of crosstalks of multiple epigenetic mechanisms such as DNA methylation and histone modifications, chromatin structure change, and untranslated RNAs. So as I told, the epigenetic mechanisms today is one of the most important studies to be carried out in relation to the diabetic one. So what are the genes? Are silent, in what condition the genes are activating, are regulating, and in what condition the genes are suppressing. So these conditions we need to understand, and the factors responsible for these regulations and expressions, if we understand, probably we can resolve the issues for diabetic management. And in many cases today now, what people are practicing is there is a reversal possibility of diabetic. Yes, definitely there is a reversible. There is a this diabetic reversal is possible because of the involvement of epigenetic mechanisms here. So we can modify, make the suitable changes in the diet, suitable changes in the exercise, suitable changes in the environment, and make the genes which are involved in this and make the genes to activate and bring back to the normal state. And this is happening, and there are some food, there are some plants, and there are some exercises involved, and create the suitable environment.
Blackboard.
And we can bring back the diabetic condition to normal conditions. And this is in practice, people are following this, but not in the name of epigenetic, but in the name of some food and exercise. Then we have another one is self-healing power of human body. This, since thousands of years, we people, particularly in India, we follow, we understand that self-healing power is there in our human body. So human body has capacity to heal on its own like other organisms. During 18th century, nosology arose and directed the medical care system towards physical structure based medicines. So the medicines which are available today, and those medicines are developed during 18th century, and before 18th centuries we had all self-belief and plant treatment, Ayurveda and many other things were available. Now the physical structures based medicines are available, and these medicines are based on the examinations or pathological anatomy, became pain diagnostic and basic for the treatment. Based on the physical structure now, we are able to identify the disease and treat the disease. This is another very important, which we follow, that the circadian rhythm. In circadian rhythm, in our biological cycle, we have different hours, our system respond to the rhythm in different way. Say for example, in the midnight, the 2 o'clock deepest sleep. That means in our body, so we say, our ancestors says that we have to take food at in the morning, around before 9 o'clock or before 10 o'clock we have to take food. Then afternoon lunch 1 or 2. Then in the evening between 6 to 7 we have to take the food, and after that we should not take food. And at 10 or 9 o'clock we should go to the bed and take rest for up to 4 to 5 hours, up to 3 o'clock or 4 o'clock. So that's very important one. So I think we are not following this rhythm or the circadian rhythm or this kind of clock. So we don't have any time for food. Anytime, whenever we feel hungry, we take the food. Whenever feel thirsty, we drink the water. And even the sleep disorders or sleep is also not appropriately we are following. And that is one of the most important one for the metabolic disorders. As per some literature, it is mentioned that we should sleep at 9 o'clock and get up in the morning 3 o'clock. And from 3 o'clock, during this period, all body detoxification mechanism occurs. And if you don't sleep during that time and be active physically, then probably that detoxification process will be destroyed or disturbed. And once at 3 o'clock, once the detoxification process completes, then there will be blood circulations or the blood circulation to different cells. And the blood circulation speed will be maximum during 3 to 4 o'clock in the morning. And that is one of the reasons, if any blood vessel is blocked, then the rate of blood movement increase will disturb, and heart attack will be more during that time. So I think this circadian rhythm we need to follow. Then, as I told, the another important is the gut microbes. So gut microbes are almost disturbed everywhere because of so many factors that we discussed. One is the food, another one is the pollutant, third one is the physical exercise. All these parameters or factors are responsible for disturbing the gut microbes in our intestine. And they are involved in nutrition metabolism, xenobiotics and drug metabolism, maintenance of structural integrity of the gut, then immunomodulation, very importantly, and they protect from pathogens. We have one more thing is autophagy. Autophagy is one mechanism which we follow in our traditional systems. We believe that our body cures our diseases naturally. So in the year 1974, The scientist Christian Duve identified this mechanism as the autophagy, and got Nobel Prize in the year 1974 for this autophagy, where our body cells, the injured cells or cancer cells or toxic cells or all defected cells, they get killed by our body's mechanisms. Autophagy, self-killing or self-eating we call, and he identified the process in which a cell eats its own content and used as a food or nutrients. And for this mechanism, he got the Nobel Prize. Okay, so all blockages, infections, substances, toxins, cancer cells, all these cells, different types of cells, they get destroyed in the body, and they used the content of those cells used as nutrients for the healthy cell. Then one more person is Yoshinori. He is from Japan, Japanese scientist. He also got Nobel Prize on this mechanism at this aspects during 2016, and he identified the lysosomes. And lysosomes plays a very, very important role in the removal of the debris or unwanted cells or defected cells from body, and use them as a nutrient. And his important contribution is all these mechanisms, they get active only under stress conditions or stress environment. If there is stress environment in the cell, then only autophagy will be activated. Otherwise, if we continue intake, we go on taking the food or intake or whatever nutrients, then this mechanism will not be activated. So that is, I think, probably in our culture, even Indian culture, we follow this one. Sometimes we have to have empty stomach, leave it for some time, and allow the lysosomes to remove the debris from the body. And for this mechanism, he also got the Nobel Prize, cellular repair and reduce oxidative stress. There is it's a sequential reaction where the lysosomes degrade the infected cells and use them as the source of food. So this I wanted to discuss with you. If anything is there, you can ask. Thank you very much for giving me this opportunity to share some of my ideas with you all. Thank you very much.